Hi, my name is Marie Neidham, and I'm a professor at Soka University of America. In this video, I'm going to discuss the taxonomy of Order Aplousobranchia in the class Ascidiacea. And the class Ascidiacea are the ascidians, commonly known as the sea squirts. They are invertebrate chordates. I'd like to acknowledge Gretchen Lambert, Francois and Claude Monio, and Rosanna Rocha for image and information. In order to determine which order you have of a particular ascidian that you might have collected in the field or you might have in the lab or in a museum, you want to think about if it's colonial. All of the Aplousobranchia are colonial, except for what I'll talk about in just 1 minute, the solitary Diazonidae. So again, if you have a colonial ascidian, and you've determined that it's an Aplousobranch, all of the Aplousobranch ascidians are colonial with this one exception. Here's the family Diazonidae, which some taxonomists actually put in the Phylum. So worms, which is the taxonomic authority, those editors put this family into the Aplousobranchia, so I will do the same. And this family Diazonidae has one genus with solitary species in it. And this genus is called Ropalia. Here's a photograph of a Ropalia abdominalis. Here's a really nice diagram from Barrel 1936 of this family Diazonidae, excuse me. And what's unique about this particular family is the solitary individuals with two body divisions. So if you've seen the earlier videos that I've put on this channel, two body divisions is restricted to colonial Aplousobranchs. This is a solitary Aplousobranch with two body divisions. If you look at this diagram, Ropalia, which is a solitary species, genus, is on the left, and then Diazona, which is a colonial genus, is on the right. But look how similar those two individuals are. So one is solitary, one is colonial, but incredibly similar as far as the morphological structure of the body goes, two body divisions. In Aplousobranchs, in terms of external characteristics, the zooids are embedded in a common tunic. Let's start with the right-hand side. That photograph is of a zooid that's been removed from the tunic. So this is what one individual zooid looks like. It's genetically identical to all the other zooids in the colony. In the center, we've zoomed out a little bit away from that single zooid to many different zooids. What you're looking at with those holes are the open oral siphons of each individual zooid. You can't see the atrial siphons because they are underneath the surface of the tunic. And then on the left photograph, zooming out even more, this is the whole colony. Zooids are embedded in a common tunic in the order Aplousobranchia with a few exceptions. Family Clavolinidae and sometimes in the family Euherdmanidae. The photograph on the left is Clavolina huntsmani, the light bulb tunicate, and the zooids are not embedded in a common tunic. They are connected to each other via stolons at the base of each zooid. So each zooid is visible and distinct, and it's not embedded in a tunic that would make each zooid difficult to pick out from the others. And then this is on the right is a photograph from iNaturalist with some members of the family Euherdmanidae. So each one of those finger-like structures is a zooid, and again they're connected to each other at their base, but they're separate at the anterior portions of each zooid. Internal characteristics of Aplousobranch zooids, 2 or 3 body sections. So on the left is a diagram of two body sections of an Aplousobranchioid, the thorax and the abdomen. The thorax is anterior, contains the oral and atrial siphons, as well as the branchial sac. So you can see those holes there are the stigmata, which are part of the branchial sac. And then in the posterior end of the animal is the abdomen, where the stomach and digestive system are, as well as the gonads, the ovaries and the testes. Also, in the abdominal part of a two-part zooid, but starting in the thorax, is what we might call a brood pouch, one of the many ways that sexually reproducing zooids keep their embryos until they emerge and hatch as larvae. So that's represented in the left diagram by the BP signal there. So those are embryos representing embryos in the brood pouch, and that brood pouch, even though it starts in the thorax, is most closely identified with the abdomen in terms of its physical location. There are other Aplousobranchioids that have three body sections. In this case, the thorax, the abdomen, and the post-abdomen. In this case, the thorax is still containing the oral and atrial siphons as well as the branchial sac. The abdomen contains only the digestive system, and the post-abdomen contains only the ovaries and testes. So in a three-body section zooid, the digestive system would be separate from the gonads. 2 and 3 body divisions are a very useful taxonomic character, and I will discuss 6 families that have 2 body divisions and 4 additional families that have 3 body divisions. First, the 6 families that have 2 body divisions. In order to separate these families in the order Aplousobranchia, I tend to focus on how many rows of stigmata, which are again making up the branchial sac. The branchial sac is what allows the animal to feed and to respire, to gather oxygen. Usually many more than 6 stigmatal rows. There are 3 families represented: Clavellinidae, Diazonidae, and Stomozoidae. 3 to 6 stigmatal rows, and that's not a usually, that's an always 3 to 6. Family Didymnidae, Holozoidae, or Polycetoridae. On the left diagram here is an example of Clavellinidae, so many more than 6 stigmatal rows. You can count them. In the middle, that's a representative of Didymnidae. It has 4 stigmatal rows. And on the right, Polycetoridae has, in this case, this particular Polycetoridae has 3 stigmatal rows. If the family is in the Aplousobranch and it has more than 6 stigmatal rows, the families that I will discuss today are Clavellinidae, Diazonidae, and Stomozoidae. Clavellinidae, the zooids are connected by basal stolons. So they are separate from each other at the anterior end and connected at the posterior end by this stolonic network, which you can see in this diagram that's in the middle of the slide. At the posterior end of the animal, those projections are stolons, and then you can see on the right side of those projections, it gets a little bit long and thinner, moves to the right, and you can see the beginning of a new zooid budding. So these zooids are genetically identical to each other. Also in the Clavellinidae, the zooids have bodies that are very elongated. So I've given you a diagram here of Pycnoclavella. You can see the extremely elongated zooid. And then just to the right of that, I wanted to show you what the animal looks like in the field, what the colony looks like in the field. Diazonidae we already talked about a little bit. The solitaries and the colonials have the 2 body parts, and unusually so in the solitaries. In this particular family, we're looking for branch longitudinal muscles, and I couldn't find a good diagram or a dissection of the branching longitudinal muscles, but what I could find in this Barrell 1936 diagram was a drawing of the muscles oblique with respect to the endostyle. So the endostyle is going to be represented on the right side of this diagram. And oblique as a term means neither horizontal nor vertical to, right? Neither perpendicular to nor parallel to. So the longitudinal muscles are going to be most visible in the thorax, and they start at the posterior end of this thorax and they radiate out in this fan shape. So they are not parallel to the endostyle, and they're not perpendicular. On the right, that photograph is Diazona violacea, which is a common species of Diazona in Europe. The third family in the Aplousobranchia that has many more than 6 stigmatal rows is the Stomozoidae. And this family was erected by Patricia Cott, and some other taxonomists think that maybe this family should be absorbed into making it not its own family. So maybe the taxonomy will change in the future. And what Patricia Cott noticed as a justification for raising this to family level was that the siphons each have 6 lobed margins. So if you're able to look at that diagram towards the right of the slide, you can see that the oral and atrial siphons both have lobes or folds with these kind of frilly margins on them. So this is Stomozoidae. If there's an Aplousobranch that you found that has between 3 and 6 stigmatal rows in each zooid, the choices are Didymnidae, Holozoidae, and Polyctoridae. Didymnidae is a huge family and is very understudied, so there may be hundreds of more species than we currently have described. A characteristic of the family Didymnidae is during asexual reproduction, there are double buds at the junction between the thorax and the abdomen. So in this series of photographs from Skold et al. 2011 from Diplozona listerianum, I've put an arrow where bud number 1 is in photograph A, and then in photograph B, I've continued to label bud number 1. In photograph C, you can see bud number 1 is now half the size of the adult zooid, and then bud number 2 is just starting to develop. And these buds emerge from the intersection, the place where the thorax and the abdomen meet. So this is unique to the Didymnidae, and it's again, buds are part of asexual reproduction of ascidians. Also, most of the Didymnidae have calcium carbonate spicules in the tunic, which you can see in this photograph from Rosanna Rocha over on the right. There are 6 genera in the family Didymnidae, so I'm going to walk you through how you might tell between these 6 genera in the Didymnidae. Diplozoma and Lysoclinum both have a straight sperm duct. So the sperm duct in this particular diagram starts, if you see the posterior end of the zooid, there's 2 testes. So they're paired, they look very similar. They're oval shaped and they have lots of little black dots inside of them. Those are the testes. And then in between the 2 testes, you see the sperm duct, which is this duct, this long line that emerges from the testes and goes all the way to the atrial opening so the sperm can be released. That is the sperm duct. And in these 2 genera, it's straight, as you can see in this diagram. How do you tell the difference between a diplosoma and a lysoclinum, given that they both have straight sperm ducts? Diplosoma is unusual in the Didymnidae for having no spicules, and it very often has slimy and extremely soft colonies. These are both photographs of diplosoma. Lysoclinum, in contrast, has spicules, so these calcium carbonate structures inside of the tunic. On the left is a picture of a lysoclinum colony. In the middle, examples of the spicules, and on the right, an example of what the zooid looks like, again with a straight sperm duct. Four other genera in the Didymnidae all have coiled sperm ducts: Didymnum, Leptoclinides, Polysyncreton, and Trididymnum. And in this diagram here, you can see on the left side of the diagram this coiled sperm duct rolled up like you might roll up a garden hose or a pastry. And this is going to be shared characteristic for these four genera. Underneath that coil is where the testes is in some cases. So the testes are basically wrapped in the sperm duct. How to tell the difference if you have a coiled sperm duct and there's four possible genera. Didymnum, photograph on the on the left of Didymnum ligulum. The testes are undivided, meaning they're in one large lobe, or they're in two lobes. This is a diagram on the right of Didymnum ligulum, and you can see that I've put an arrow to the single testes, undivided, only a single lobe. In Leptoclinides, photographs on the left, it's the atrial siphon that you're going to look for. It's tubular and long, and in this case it points downwards, points posterior in the structure of the animal. In Polysyncreton, a photo of the colony on the left, the atrial aperture, which is where the waste and the sperm and the eggs are released, and the water after the nutrients have been taken out. That's the atrial aperture. It has an anterior lip. So a projection on the anterior portion of that atrial area that projects out past the zooid. And that would be a characteristic of Polysyncreton. And finally, you have an easy one here, Trididymnum, tri for three. This is the only Didymnidae genus with 3 stigmatal rows. All the rest of them have 4 stigmatal rows. I'm now going to move on to a different family in the Aplousobranchia. So just to orient you, I'm explaining the difference between the families in the Aplousobranchia where the zooids have 3 to 6 stigmatal rows. I talked about the Didymnidae just now. I'm also now going to move on to the Holozoidae. In this particular family, the embryos are incubated in a thoracic pouch. Morphologically, these colonies can be quite different from each other inside the Holozoidae, so I gave you photographs to represent some of that diversity. Sigillina on the left and Dystaplia occidentalis with this capitate or mushroom-like colony morphology. In the diagram on the right, you can see that thoracic pouch, right? So it is separate from the rest of the body. It's connected to the atrial opening in the thorax by a very thin connector, and then it's a long, thin structure moving posteriorly down past the stomach, and contains the embryos. So in sexually reproductive colonies, that's where the embryos are brooded until they hatch as larvae. Another family that has 3 to 6 stigmatal rows is the Polycytoridae. And the way to tell this particular family is the siphons are tubular and they have 6 rounded lobes. So that diagram you can see both in the oral and atrial siphons, they're more tubular than the other families and they have 6 rounded lobes. There's 2 common genera in the Polyctoridae. The first is Eudistoma, and the way to tell this one is that it has 3 stigmatal rows. There's a photograph of Eudistoma olivaceum from Rosana rocha, a very common Eudistoma from the Caribbean. And again, the zooid has 3 stigmatal rows. As opposed to Cystidites, the other common genus in the Polyctoridae family, has 4 stigmatal rows, but also it has very distinctive discoid spicules, so circular spicules covering the abdomen, as shown in this drawing here. You might also remember from earlier in these videos that Cystidites also has horny plates covering the test. I'd like to now move on to families with 3 body divisions. There are 4 common families: Euherdmanidae, Polyclinidae, and Pseudodistomidae ritterellidae. And the 3 body divisions would be the thorax, the abdomen, and the post-abdomen. First, the Euherdmanidae. In the Euherdmanidae, the zooids are often separated from each other as opposed to embedded in a common tunic. This photo is from iNaturalist, and each one of these structures that you see is a zooid. They're covered in sand. They live in the intertidal, and you can count each zooid. They're not obscured inside a common tunic. In the Polyclinidae, the atrial siphons of the zooids don't open directly to the surface of the colony. They open to a common cloaca, which is Latin for sewer. And I've given you arrows there to see those large openings in the colony. Those are cloaca. The smaller openings are the oral siphons corresponding to each of the zooids. There are 2 common genera in the Polyclinidae. One is the Aplydium, and it has a folded stomach or plicated stomach. This nice drawing from Montesanto et al. In 2019, in the center you see the diagram of the stomach. It's got an ST label and there's folds in the stomach. You can also see photographs of the stomach, yellow just to the left of the diagram and stained purple just to the right of the diagram. So there are folds in the stomach in Aplydium. As opposed to the other common genus in the Polyclinidae, which is the Polyclinum, the stomachs are smooth, no plications, no folds. In the center is a diagram of a Polyclinum zooid, and the stomach is represented by the label ST. You can see it's smooth, there are no folds. On the right is the colonies of Polyclinum constellatum. In the 3 body division families of Order Aplousobranchia, we have 2 more families to discuss, the Pseudodistomidae and the ritterellidae. Both of these families have atrial siphons of the zooids open to the colony surface. So no cloaca. Every zooid has both an oral and atrial siphon that open directly onto the surface of the colony. How to tell the difference between these families, the Pseudodistomidae and the ritterellidae: the number of stigmatal rows. Pseudodistomidae represented on the left here, both a picture of a colony and the zooid, 3 stigmatal rows. Ritterellidae represented in the right of this slide, more than 3 stigmatal rows.